Hello student, welcome to Anu's classes. This is my second video on the topic blood. In my previous video, I have discussed about what is blood, component of blood, function of different component of blood and structure of blood. So let's proceed to this video with another important topic of blood, that is blood group. In the world, there are generally two types of blood group which are most widely used all over the world. These are ABO blood group and RH blood group. And these blood groups are decided according to the antigen present on the surface of the RBC. First come to the ABO blood group. Under ABO blood group, there are mainly four types of blood group, that are blood group A, blood group B, blood group AB and blood group O. So under ABO blood group, there are total four types of blood group. So, and these blood groups are decided according to the antigen present on the surface of the RBC. So, suppose this is the cell of RBC, and on the surface of this RBC, I am showing with two different color. I am showing here one green antigen. This is suppose antigen A. So, this is antigen A, and this blood group is named as A blood group. So now come for the another RBC. Suppose this is another RBC. Here I am drawing the antigen in a red color. So this antigen is named as suppose B. So this blood group is still B blood group. Next another RBC, and here both antigen are present. This is A antigen and this is B antigen. So this blood group will be named as AB blood group. Again another RBC where no antigen is present. So this type of blood group will be called 0, means O blood group. So these four types of blood group are found under ABO grouping. Again in the A blood group where antigen is A, their antibody will be always B. Means in A antigen their antibody should be small b. When B antigen, their antibody should be always A. Then AB, here two antigen is present. That's why here no antibody will be present. And this is O blood group where no antigen is present. There both type of antibody, means A and B are present. Always remember that the antigen and antibody should be always opposite, because same antigen and same antibody always coagulate, means they react with each other and clotting will occur, which will be fatal for the person. So if antigen will be A, antibody will be B. If antigen will be B, antibody will be A. And here the two antigen, therefore it is named as AB blood group, no antigen, and O blood group with no antigen but antibody will be A and B. Now come to the blood donation process. So during the time of blood donation, the antigen of the donor and the antibody of the recipient should be always matched. Otherwise, it will be fatal for the person. Suppose a person who have A blood group, and so its antibody will be B. So A blood group, and if he is donated blood group B, suppose this is RBC of blood group, this is B blood group, antigen will be A. So B blood group means antigen will be B. So what happened in this case, this B antibody react with the B antigen and they will interact with each other, and as a result they coagulate and clotting will be takes place, will be fatal for the person. That's why the antigen and antibody should not be same during the blood donation. So this is I am showing in the chart. Look, this is blood group A, which says A antigen, B antibody, B antigen, A antibody, AB, no antibody, O, A and B antibody. So blood group O is called universal donor. Why it is called universal donor? It is called universal donor since it has no antigen. So no antibody can react with this blood group. So it can donate blood group to any blood group. That's why it is called universal donor. At the same time, AB is called universal recipient. Why? Since AB blood group has no antibody, so any antigen can enter in this blood group. Because it has no antibody, so it cannot react with any antigen. That's why it can receive blood from any blood group. That's why AB is called universal recipient, whereas O is called universal donor. Now come to the another type of blood grouping, that is RH blood grouping. So RH blood grouping means, suppose this is A blood group with antigen A, another antigen, if RH antigen is present, this type of blood group is called RH positive, means this blood group is named as A positive. Suppose if RH antigen is present on B blood RBC, then this blood group is called B positive blood group. Like that, if suppose this is a blood group A, so antigen will be B, antibody will be B and antigen will be N, but RH factor is absent, then this blood group is named as A negative blood group. So if RH factor is present on the surface of the RBC, then that blood group is called RH positive, and if RH factor is absent, then that blood group is called RH negative blood group. So now if an RH negative person, this is RH negative person, here RH antigen is absent, is exposed to RH positive blood, this is RH positive blood, this is RH antigen, then in this person's blood there will form a specific antibody against this RH antigen. Since though this is A negative, this is A positive blood, still here RH is present, here RH is absent. So in this blood group, one antibody will be formed which is against this RH antigen. Therefore, RH groups should also be matched before the transfusion. A special type of RH incompatibility has been observed between the RH negative mother and RH positive baby. It means if a RH negative mother carry a baby which has RH positive blood, then this type of incompatibility has been observed. It means suppose this is the body of the mother who is RH negative and this yellow portion indicating the fetus who is RH positive. It means it's RBC with RH antigen. And this pink color is the maternal circulation, this blue color is the placenta. So in case of first pregnancy, no complication which arises as the blood of fetus and the blood of mother will be completely well separated by presence of this placenta. But during the time of first delivery, there is maximum chances of exposure of the RBC of fetus with the mixing, means there is maximum chance of mixing of the RBC of the fetus with the RBC of the mother or blood of the mother. And as a result, the mother's body start producing antibody against this RH antigen. Means due to the presence of this RH antigen, it activates the RH specific beta cell, B cell, which are present in the mother's blood. And this specific B lymphocyte cell starts producing two types of cell. One is memory cell, another is plasma cell. This plasma cell will produce a large number of antibody which are called anti-RH IgM. These are larger in size, and this memory cell will memorize the characteristic of this RH antigen which activate their parent cell, and they will remain in the blood for decades and circulate within the blood, and they will act as a previously infected antigen. So in the next time of next pregnancy, means second pregnancy, when again the RH negative mother carry a RH positive baby, then if some blood of the baby is mixed with the blood of the mother, then immediately, as in the mother's body already the memory cell which act against this RH antigen and the antibody are present in mother's cell, so immediately this memory cell will start producing another type of antibody against this RH antigen which are called anti-RH IgG, which are very smaller in size, and due to their smaller size they can penetrate the wall of the placenta and they can enter into the bloodstream of the fetus, where they start destroying the RBC of the fetus, and which cause maybe fatal to the fetus or which could cause severe anemia or jaundice to the baby. And this condition is called erythroblastosis fetalis. And this type of situation can be avoided by administering anti-RH antibody to the mothers immediately after the delivery of the first child. Now come to the another important topic, that is coagulation of blood or blood clotting. So blood clotting is the process which prevents the excessive loss of blood from the body. To understand this mechanism, we have to go through this flow chart. So this is a injured portion and these are the injured tissue. This injured tissue will release thromboplastin. Thromboplastin are a mixture of phospholipid and tissue, and this injured tissue also activate the platelets present in the blood. As a result, the platelets come and accumulate in this injured portion. That platelets also release the thromboplastin. These thromboplastins, in the presence of calcium ion, they produced a enzyme which is known as thrombokinase. And then this thrombokinase enzyme help, and as a result the prothrombin, which is a protein, are converted into thrombin, which will a type of enzyme and which catalyzes, and as a result it's helped in the formation of fibrin, or it convert the fibrinogen to the fibrin. These fibrinogen are soluble, whereas these fibrin are insoluble fiber. And the damaged and the dead tissue of blood cell will be trapped in this fibrin, and as a result clotting is done. So in this process we have to remember the term, this is thromboplastin, which are released by injured tissue and the platelets. Then thrombokinase, this is an enzyme which converted this prothrombin to the thrombin, and this thrombin again converted the fibrinogen, means this soluble product, into fibrin insoluble fiber, where the damaged blood tissue are trapped. In the whole process of blood clotting, total 13 factors are involved, and calcium plays an important role here. And the order of blood clotting we can write simply like that: this thromboplastin's formation, then conversion of prothrombin to thrombin, then conversion of fibrinogen to fibrin, then formation of clot. So this is the process of blood clotting. In my next video, I will discuss on another important topic of the chapter, that is lymph and human circulatory system. Thank you.